Okay, welcome back. We are talking about Chapter 9 material, and we just finished the process of aerobic cellular respiration and looking at how cells can take nutrients like glucose and oxidize them and use that energy to make ATP. It's complicated, it's busy, it's crazy, right? But we just finished it. So now we're ready to move on. I'm going to hit our PowerPoint slides, and we'll see what's next. This should be the very last video on Chapter 9, believe it or not. And we're going to cover fermentation and some other wrap-up materials for Chapter 9. And so we're really at slide 32 at this point. And I'm going to open that up slowly. Okay. So I have a few extra things that I decided not to cover. We're not going to we don't have to cover this. It just asks the question, do bacteria also use aerobic cellular respiration? Can they? And they do. The short answer is yes, you bet. They don't have mitochondria, but they have membranes in which they can do aerobic cellular respiration. Some bacteria don't, but others do. Here's where we're really going to start. We've covered aerobic cellular respiration. That's one mechanism for that cells use to make ATP. Another mechanism is called fermentation. Fermentation is a mechanism of harvesting energy, making ATP, without the use of oxygen. Okay? So fermentation is not going to use oxygen. And there are different kinds of fermentation, or different types. This is the only slide with anything molecular on it. We'll talk about this, but I want to introduce fermentation. I added an extra slide. You're going to just have to jot down some notes here about fermentation. It's called fermentation. So yeah, that's spelled incorrectly. So fermentation, here's a few basic notes about them. In fermentation, cells that use this mechanism of making ATP also oxidize glucose. They use nutrients like glucose or other kinds of foods, and they oxidize it. Okay. They make ATP by substrate level phosphorylation, and that's it. They don't use that whole process of oxidative phosphorylation. They don't have an electron transport chain and chemiosmosis happening. They simply make ATP by substrate level phosphorylation. This isn't very efficient. In fermentation, one molecule of glucose can be used to make about 2 ATPs. Aerobic cellular respiration is much more efficient. Remember, one molecule of glucose made 30 to 32 ATPs. So when organisms use fermentation, it's not super efficient. Some organisms don't care. It works for them. Right? And this is also interesting. No oxygen is required for this mechanism. Researchers believe that fermentation probably evolved before aerobic cellular respiration, probably during the time in which there really wasn't much atmospheric oxygen available, before plants started photosynthesizing. So many microorganisms, bacteria and archaea, can use fermentation and fermentation only to survive and thrive. There are different types of fermentation. We're going to take a look briefly at 2 of them. Some cells can survive entirely by fermentation. Other cells use fermentation when there isn't much oxygen available, or use it as kind of a supplementary way to make ATP. But the kicker is, in fermentation, just like in aerobic cellular respiration, NAD+, remember our electron shuttler, must be regenerated. NAD+ has to be regenerated in aerobic cellular respiration also, so that it can continue. In fermentation, it must be regenerated too. So now I'm going back. Let's take a look at the 2 types of fermentation that I've posted here. Okay? One of them is called alcohol fermentation. The other is lactic acid fermentation. Okay. Lots of organisms can use what's called alcohol fermentation. Bacteria, fungi, plant cells. And in alcohol fermentation, let's take a look at the picture. We start with glucose, okay? And glucose goes through glycolysis. Oh, those 10 steps of glycolysis to make 2 pyruvates. And some ATP by substrate level phosphorylation. Remember that during glycolysis, 2 NAD plus molecules are reduced to NADH. Okay? In order to continue this process of glycolysis, a cell needs to regenerate NAD plus so that it can be the oxidizing agent to oxidize glucose again and again and again. Fermentation takes the products of glycolysis and uses them to regenerate NAD plus. In this case, in alcohol fermentation, the 2 pyruvate molecules first lose some carbon dioxide, and then the acid aldehyde molecule that's left over is reduced to ethanol, picks up electrons from NADH. Therefore, NAD plus is regenerated so that glycolysis can happen again. So fermentation takes advantage of the 10 steps of glycolysis to make ATP, but then regenerates NAD plus in some way. In this case, the pyruvate molecules are first converted to acid aldehyde, and then acid aldehyde is reduced to an alcohol. And that's why it's called alcohol fermentation, right? The type of enzyme used to get this process done has to be an enzyme used in catalyzing redox reactions, because this is a redox reaction. It's a dehydrogenase. Now, fungi like yeast can ferment without oxygen, and we use yeast, right, during baking. We throw it into our bread dough. Why do we use it? Well, we use it to help the bread dough rise. Why does the bread dough rise? As fermentation is happening, so you have to have some sugar in the bread dough too, right? The yeast are using the sugar, they're fermenting, they're going through the steps of glycolysis, fermenting, and as they ferment, they release carbon dioxide bubbles. Those bubbles of gas kind of bubble up through the dough and cause it to rise. The alcohol that's produced is burned off during baking. Anyway. Lactic acid fermentation. Oh, by the way, one more thing. We made 2 ATP from 1 molecule of glucose. Not super efficient. Lactic acid fermentation looks very similar. Many organisms like bacteria, fungi, and animal cells use lactic acid fermentation. We use lactic acid fermentation in our muscle cells. So if you're trying to outrun a saber-toothed tiger, you need to be sprinting as fast as you can, right? And so you're aerobically respiring, you're taking in oxygen, you're running as fast as you can, your muscles are making ATP by aerobic respiration, but they need more. They need more strength and more power to outrun that saber-toothed tiger. I don't know, it's probably hopeless anyway, right? But then our cells can add lactic acid fermentation. Doing lactic acid fermentation makes more ATP so that our muscles can be powered or fueled even more. So what we do with lactic acid fermentation is we add it to aerobic cellular respiration, so that increases the amount of ATP we can make. Some organisms can survive solely on lactic acid fermentation. We certainly can. Okay, well, what does this look like? Very similar, right? Glucose is converted into 2 pyruvates through the process of glycolysis. We're making 2 ATP along the way, and NAD plus is the oxidizing agent that oxidizes glucose. It becomes NADH, right? And now in the process of fermentation, pyruvate is reduced to lactate or lactic acid. Right? This is happening in the muscles, and a dehydrogenase enzyme makes this happen. What that does is it regenerates NAD+ again, so that glucose can go through, more fermentation can take place, really. The lactic acid that is produced in the muscle cells is shipped off to the liver, where it's reconverted into pyruvate. Sometimes people say that lactic acid fermentation leads to really sore muscles. So if you're sprinting a 400-meter run, your muscles can be really sore at the moment. That's the kind of lactic acid soreness I'm talking about. Chances are that your muscles can get really sore during lactic acid fermentation because the pH in your muscle cells drops. It becomes more acidic as a result of lactic acid fermentation. And that can trigger your pain neurons, and that can trigger pain. Anyway, it's kind of interesting. We certainly don't use lactic acid fermentation as our only mechanism of making ATP. It's not super efficient. Okay. This is just another slide looking at when fermentation happens, because some organisms are what we call facultative anaerobes. We're aerobic. We need oxygen to survive. Some organisms are anaerobic. They get poisoned by oxygen. They can only do things like fermentation. Other organisms are called facultative anaerobes. When oxygen is present, yay, they get to do cellular respiration. But when no oxygen is present, they can do fermentation and survive. Okay? So that's kind of a drawing of what happens when oxygen is present versus when it's not in some organisms. Right? Okay. A couple more topics, and they're short. Anaerobic respiration is touched on in your textbook. I'm not going to focus on this, so I'm going to kind of scratch it out. Just so you know, anaerobic respiration is not fermentation. Anaerobic respiration doesn't use oxygen. It's a way to make ATP without the use of oxygen, but it does use, the word respiration here is important, it does use an electron transport chain. In anaerobic respiration, an organism uses electron transport chain, but the final acceptor of electrons is not oxygen. It's something else. So it's still respiration. We still see oxidative phosphorylation happening. It's not fermentation, but there's no oxygen involved. Some cool bacteria like E. coli can use anaerobic respiration. We will touch on this topic. Alternate fuels for cellular respiration. Can cells use different kinds of molecules to make ATP? And so far, all we've talked about is glucose, Glucose is the initial substrate that cells use to make ATP by aerobic cellular respiration. Can they use anything else? Can we use more complex carbohydrates, like grains and starches? Can we use fats? Can we use proteins? Your gut reaction to this question is, yes, of course we can. That's the right answer. Yes, of course we can. Our cells can use a whole variety of molecules to make ATP by aerobic cellular respiration. This is a diagram that shows us that, we can use proteins or carbohydrates or fats in the process of aerobic cellular respiration. We just use them in slightly different ways. So if we eat, a bagel for breakfast, we're going to have some complex carbohydrates that need to be broken down into simpler sugars, like glucose and fructose, and they can go through the process of, the whole process of aerobic cellular respiration. They can be inserted into glycolysis, if you will. If we use proteins, what about the eggs you ate for breakfast, right? What happens to the protein in those eggs? Well, the proteins get broken down into amino acids. They're deaminated, so nitrogen is removed, and what's left over can then be tapped into. There's energy in those amino acids. Keep that. That can be used, be converted into pyruvate or acetyl-CoA or some other intermediate in the citric acid cycle. and they can be tapped into to make ATP. Yeah. Right. If you eat a diet of only protein and fat, fats. Let's talk about fats. Fats are over here. Fats are really good sources of energy, right? Fats can be broken down into components. Glycerol are part of triacylglycerides. They can be converted into glyceraldehyde 3-phosphate. That's a part of glycolysis, used in the rest of the cycle. Fatty acids, they're rich in energy. They can be used to be converted into acetyl-CoA and then used in the citric acid cycle. Absolutely, all of these foods can be used for this process. Okay. If you eat a diet of only proteins and only fats, will your body starve itself from, will it not be able to do aerobic cellular respiration correctly, or will it have some problems? It can. I would not recommend eating a diet that's only fats and proteins, but typically our bodies can adjust and can work with that. If you have diabetes, your body can't use the sugars that it takes in, and that can be dangerous. If it can't use any sugars that it takes in, then the body has to use the fats and proteins, and sometimes the blood gets too acidic as a result of that. And that can lead to problems like comas and such. And so that's why it's so important for people who have diabetes to regulate their diabetes with insulin intake or just monitoring their diet so that they don't have such problems. One more topic yet to go, and I'll be right back on this topic.